Hi everyone, welcome back to the Science Box. So today I'm going to be telling you everything you need to know for the photosynthesis chapter of AQA A Level Biology. The timeline's in the description, so if there's a particular part you're stuck on, feel free to just skip to there. Photosynthesis happens in the chloroplast, so we're first going to look at the structure of a chloroplast. So if we cut a chloroplast in half, it'd look a bit like this. Firstly, we have an inner membrane and an outer membrane. Inside that is the fluid-filled space called the stroma. Also inside the chloroplast are stacks of disc-like structures. The whole stack of discs is called a grana, and each individual disc is called a thylakoid. The membrane of each of these discs is the thylakoid membrane, and the space inside the disc is called the lumen. Now, photosynthesis can be broken down into two main stages. The first stage is the light-dependent reaction, and the second stage is the light-independent reaction, also known as the Calvin cycle. We're going to start with the light-dependent reaction, which happens in the thylakoid membrane. So this is our setup for the light-dependent reaction. These things called photosystems are basically just containers for chlorophyll molecules, which are all these green dots, and the black dots inside them are electrons. So step number 1, light strikes chlorophyll molecules in photosystem 2, transferring energy to 2 electrons. The energy level of these electrons is elevated, so they leave the photosystem, and as a result, the chlorophyll molecule gets ionized. So this process of the electrons getting energy and leaving the chlorophyll is called photoionization. Step 2, the electrons move through the protein carriers, releasing energy, allowing for ATP formation. Step 3, light strikes chlorophyll molecules in photosystem 1, exciting a pair of electrons in those chlorophyll molecules. These electrons move through the electron transport chain, releasing energy to form ATP, and are eventually used to reduce NADP to NADPH. These electrons lost are replaced by those from photosystem 2. Electrons lost from photosystem 2 are replaced by electrons from the photolysis of water. Photolysis, made up of the words photo and lysis, is where light energy is used to split the water up into protons, electrons, and oxygen. So the equation is 2H2O goes to 4H plus 4E minus plus O2. These protons produced undergo chemiosmosis through ATP synthase, allowing for the synthesis of ATP. The protons are then used in the reduction of NADP to NADPH. In summary, our products of the light-dependent reaction are NADPH and ATP. Now, the light-independent reaction, or the Calvin cycle, happens in the stroma of the chloroplast. Step 1. Carbon dioxide binds with the specific complementary active site of the enzyme rubisco. There is also a second binding site of rubisco which ribulose bisphosphate, or RUBP, binds to. Step 3. Ribulose bisphosphate is carboxylated to form 2 molecules of glycerate 3-phosphate, or GP. Step 4. ATP and reduced NADP from the light-dependent reaction are used to reduce glycerate 3-phosphate into triose phosphate. Step 5. Some triose phosphate molecules are recycled to form RUBP, and some triose phosphate molecules are converted to useful organic substances like glucose, fructose, and glycerol. So our ultimate products of photosynthesis are glucose and oxygen. Oxygen is made in the light-dependent reaction during photolysis, and glucose is made in the light-independent reaction. Okay, now moving on to photosynthetic pigments. The two main types of chlorophyll are chlorophyll A and chlorophyll B. Chlorophyll A is the most abundant pigment, and it absorbs blue/violet light and red light. And chlorophyll B absorbs blue light and orange/red light. The leaf also contains accessory pigments which allow the leaf to optimize on the light absorbed by absorbing the wavelengths of light that chlorophyll won't. Chlorophyll is green, so it reflects green pigment rather than absorbing it, but accessory pigments called carotenoids, for example, absorb blue and green light, and xanthophyll is another one, and that absorbs yellow light. Okay, now moving on to the required practicals associated with this chapter. There are two, required practical 7 and required practical 8, and we're going to start with required practical 7, which is chlorophyll chromatography. Okay, so step 1, we extract the pigment from the leaf by crushing the leaf up in a pestle and mortar and adding a solvent to it. Then step 2, take the chromatography paper or the TLC strip and draw a line a few centimetres above the bottom of the chromatography paper in pencil. We use pencil because if we use pen, the ink would dissolve into the solvent and intervene with the pigment's chromatography. Then, using a pipette, put a spot of the pigment on the pencil line. Allow this to dry and repeat the process over and over again until the pigment dot is 3 to 5 millimeters in diameter. Then pour the solvent in the beaker and then hang the chromatography paper in the beaker so that the solvent line is below the pencil line. This is so the pigment travels upwards with the solvent rather than dissolving into the solvent at the bottom of the beaker. Next, put a lid on the beaker so that the solvent doesn't evaporate. Allow the sample to run up the paper, but take the paper out when the solvent is a few millimeters from the top, and draw a pencil line to mark where the solvent reached. Okay, now we can calculate an RF value. This is useful because we can compare this value with published data values and identify the pigment that we found. When comparing it with a data book, we need to make sure that both values are using the same solvent, because different pigments have different affinities to different solvents. The equation for the RF value is distance moved by pigment over distance moved by solvent, and we tend to measure the pigment from the middle of the pigment spot. Okay, now moving on to required practical 8. So the aim of this investigation is to investigate the effect of the addition of a named variable on the rate of the dehydrogenase enzymes in chloroplasts. The dehydrogenase enzymes catalyze the conversion of NADP to reduced NADP, also known as NADPH, and in this experiment we'll be using DCPIP, which is a redox indicator, which is blue when oxidized and colourless when reduced. It can pick up the electrons from the light-dependent reaction instead of NADP. And the named variable we'll be using in this case, as recommended in the practical handbook, is ammonium hydroxide. Ammonium hydroxide is alkaline, so may denature the dehydrogenase enzyme, but also may accept the electrons, preventing the DCPIP and dehydrogenase enzymes from doing so. Okay, so first I'm going to be talking you through the test tubes we're going to set up in the experiment. Only the last two, so test tubes 4 and 5, are the actual experiment, and the rest of them are for comparison's sake or act as controls. Okay, so test tube 1 is our standard. It contains chloroplast suspension and water. We're going to compare our experimental tubes to this tube because when the DCPIP goes colourless, due to the chlorophyll in the tube should go green, and it should be the same shade of green as in test tube 1. Test tube 2 is our first control tube. It contains chloroplast suspension, water and DCPIP, but is then covered in foil. This is to prove that the DCPIP won't decolorize without light. So that proves that in our light-dependent reaction, electrons are only released when light is available. Test tube 3 is our second control. This contains water, DCPIP, and an isolation medium, but no chloroplast solution. This is to prove that the chloroplasts are needed and that dehydrogenase is within the chloroplasts. Test tube 4 is experimental tube 1. This contains water, DCPIP and chloroplast solution. The solution is a mixture of the blue DCPIP and the green chloroplast solution, making it turquoise in colour. And then finally, test tube 5 is experimental tube 2. It contains the exact same components as test tube 4 with the addition of ammonium hydroxide. By comparing tubes 4 and 5, we can see the effect of ammonium hydroxide on the rate of dehydrogenase enzymes. Okay, so now I'm going to talk you through the method, and we're going to start by making the chloroplast suspension. So the chloroplast suspension can be made by just blending up or homogenizing some spinach leaves combined with the isolation medium. This is a bit of revision from the cells chapter actually, but the isolation medium must be cold to reduce enzyme activity that might break down the organelles, and isotonic to prevent organelles bursting or shrinking as a result of the osmotic gain or loss of water. Just a reminder that the solution we place a tissue in before cell fractionation must be these two things, but also buffered so that the pH does not fluctuate. Any change in pH could alter the structure of organelles or affect the functioning of enzymes. Anyway, next we filter the homogenate to remove unbroken cells or large pieces of debris, and then we put all the test tubes in a water bath so they're all at the same temperature. Point a light source at them and time how long it takes for test tubes 4 and 5 to turn the same colour as test tube 1. When you do this experiment, the results should show that test tube 5 takes longer to become green, the green that we're looking for, than test tube 4. This proves that ammonium hydroxide decreases the rate of dehydrogenase activity. Now, there are some limitations to this experiment. Firstly, the endpoint is subjective. We tried to accommodate this by having a standard tube to compare the colour to, but to make the results quantitative, we could have used a colorimeter, and that would have been more accurate. And then secondly, the light distribution may be uneven, so some tubes are getting more light than others. Okay, so that brings us to the end of our video. Thank you for watching. Please like and subscribe. If you like this style of teaching and would like to get tutored by me or a member of our team, you can go to our tutoring website at thesciencebox.net. I'd also really recommend our Instagram and TikTok for tiny doses of revision. It makes you feel productive even if you're just scrolling TikTok, so that's a bonus.